Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture from Kami Biology. In this video lecture, we are going to talk about nucleic acid inhibitor drugs. Fine. In the previous video lecture, if you remember, we talked about the beta-lactam antibiotic in a detail. We also talked about the cell membrane inhibitor drugs. Fine. So now this is another video lecture regarding with antibiotic drugs, and in this video lecture we are going to talk about nucleic acid inhibitor drugs. Nucleic acid inhibitors, especially in bacterial cell. So let me write. Nucleic acid inhibitor. We usually call that fluoroquinolones. Fluoroquinolones. Maybe the spelling is right or wrong, then you will check the spelling. We usually call that fluoroquinolones. Fine. Now, what is nucleic acid? Nucleic acid are those, for example, DNA and RNA are the nucleic acid. These genetic material, we also call it genetic material. These are very important for the cell dividing, or we can say for the genetic information or cell to cell information. These nucleic acid are very important. These nucleic acid are play important role in the cell division as well. Fine. So in nucleic acid, we usually focus on DNA, fine, because in then in other video lecture we will focus on RNA. But now in this video we are just focusing on DNA replication inhibitor, fine. Although in case of nucleic acid there is DNA, RNA, but here we are just focusing on DNA, fine. So here we just focusing on DNA replication inhibitor. DNA replication inhibitors. Now DNA replication is very important for cell. Due to DNA replication the cell will be divided, and due to DNA replication two daughter strand of the new DNA strand are formed by which the cells are divided, fine. If we focus on nucleic acid, then you better know, in case of central dogma, if I draw the central dogma, you better know that from DNA, if we talked about central dogma, so now from DNA, so let's suppose this is the DNA, fine. What we get RNA, and from this RNA we get protein. Let's suppose these are the protein, fine. And from this DNA sometimes we also get the other DNA, fine. So this is called replication. Now listen, from DNA when we get another DNA just like this, or they behave just like this, we call that a replication process. From DNA when we get RNA we call that transcription, and from RNA when we get proteins we call that translation, fine. So all these processes are needed for the cell survival, for the cell replication, for the cell division. All these processes, this central dogma are needed, fine. And this central dogma will be started through this DNA. So DNA are very important, fine. Now if we talk fluoroquinolones drugs, so the drugs which are involved in fluoroquinolones, these drugs are the, let me write the names. These drugs are usually the ciprofloxacin, getafloxacin, and norfloxacin, moxifloxacin, fine. So these are the fluoroquinolones drugs. These fluoroquinolones usually target the DNA replication, or we can say these fluoroquinolones will stop the DNA replication process. Once the DNA replication process are stopped, then DNA means the DNA will be not formed, and whenever the DNA is not formed, genetic information will be not transmitted through the cell or outside the cell. Inside the cell or outside the cell, the DNA, the genetic information will be not transferred. Fine. Second thing is that cell will be not replicated, or cell will be not divided. Fine. So DNA replication is very important steps, but this fluoroquinolone will target the DNA replication process. How? Now listen. Before going to explain the mechanism of action of this fluoroquinolone, so you need to understand the DNA replication. Here I will just a little bit explain that how the DNA are replicated inside the cell. I'm not going to talk in a detail about the DNA replication because it is very complex and a very detailed topic. But here in this fluoroquinolone's mechanism of action, I will just a little bit touch. Fine. So inshallah in next videos we will be talked in a detail about the DNA replication. Fine. Now how the DNA is replicated. So pay attention here. Now listen. Let's suppose this is a DNA strand. Here I will not draw the exact structure of the DNA. Fine. So it is just a simple structure of the DNA. Fine. So let's suppose this is the DNA strand. Fine. Now during replication, what actually happens, this DNA strand will be a little bit, this DNA strand will unwind their self. Means, let's suppose this one area of the DNA strand will be unwind, and usually we call it replication fork. We can say that during DNA replication, a replication fork is formed. This is a replication fork. Fine. So what happened actually in replication fork? After that, then again the DNA strand will be present, but this DNA strand is now in a supercoiling form. Fine. So this is the supercoiling. Supercoiling. Now pay attention here. During replication, what actually happened? The helicase enzyme come. Let's suppose this triangular shape represents the helicase enzyme. Fine. This helicase enzyme will come and they will unwind the DNA. When they unwind the DNA, so then this helicase will move. The helicase enzyme is going downward. Then what actually happen? They will just go downward, and they will they will break the hydrogen bond, and here they will also unwind the DNA in that area. And whenever they are going downward, or whenever they are going this direction, so they will release that they will unwind the DNA and unwind. When this enzyme, let's suppose helicase reach to that area, then after that here supercoiling will be occur. What does it mean? It means that the helicase try his best to unwind that area, but they fail. Why? Because supercoiling occur here. Here the DNA are very in a supercoiling form. Their DNA are twist around with each other, but this twist around will again coiling, and this coiling is again coiling with each other, by which the supercoiling are formed. And this supercoiling is in two forms, positive and negative. Is in two forms, positive and negative. These represent the direction of the coiling. Now this DNA strand is always in a negative supercoiling. Fine. So keep it in your mind. The DNA strand will always in a negative supercoiling. So the DNA strand is always in a negative supercoiling. So what actually happened? To release this tension, there is two main important characteristics will occur. Fine. So, or we can say there is an enzyme which come and release this tension. Once this tension is released, so again helicase will break the bond and will make a replication fork. Fine. Now what actually happened? Let me tell some other things. Once the replication forks, here DNA polymerase enzyme. So let's suppose this one represents the DNA polymerase. DNA pol. The DNA polymerase enzyme will come here and they will make a new strand which is complementary to this one strand. They will make a new strand. Fine. So here the new strand are formed. This blue represents the new strand of the DNA. Fine. So now what actually happened? And then this helicase will go downward. Then an enzyme we usually call that topoisomerase. So let me write the name. Topoisomerase. Fine. So there are many types of the topoisomerase, but in this case I will just explain two main types of the topoisomerase, and these are the topoisomerase 2 and topoisomerase 4. 2 and 4. Fine. Now what actually happened? This topoisomerase 2 will cleave this DNA strand. This topoisomerase will cleave one strand of DNA. Topoisomerase 2 is cleave one strand of the DNA, and this topoisomerase 4 cleaves and join both the strand. What does it mean? It means that this topoisomerase means the main function of this topoisomerase is to release the tension. So how it releases the tension? First mechanism of this topoisomerase enzyme is that they will cleave the strand. Let's suppose they will cleave at that area. Once they cleave at that area, so tension will be released. Supercoiling will be a little bit relaxed, or a DNA will be in a relaxed form. A little bit, not a lot. A little bit the DNA will be relaxed. And this topoisomerase 4, what actually this topoisomerase 4 do? They will cleave, let's suppose at that area they will cleave. Fine. And once they cleave at that area, then what actually happen? They will join this one side to that area, and then that area, and then that area, by which the twist form of the DNA will be occur. We can also say that. Let me remove this one. Means they will cleave some area of the DNA strand, by which the DNA strand will be in this form. So when the DNA strand in this form, so again the helicase will move in this direction. So when the helicase moves in this direction, so again replication fork is formed, again the DNA strand will be formed. Again this polymerase will reach to that area, and again another new DNA will be formed. Fine. So this is the replication. Fine. So this topoisomerase will sometime cleave the one strand of the DNA to release the tension, or this topoisomerase 4, they will cleave the DNA, one strand of the DNA, and join the strand or some part of the strand of the DNA with each other to release the tension. Fine. This topoisomerase 4 enzyme is usually called that gyrase. Fine. So this topoisomerase enzyme is actually multi-subunit. What does it mean? It means that it will be in this form. Means it can perform two functions. Fine. So this topoisomerase 4 is called as DNA gyrase, and it will do two functions. Why? Because it has multiple subunits. Now let's suppose this one site is responsible for the joining, and this one is responsible for the cleavage. Fine. So this one site will come and they will cleave some area to release the tension, and then due to this one site the enzyme will come and they will bind, they will reseal that area. Fine. So that's all about the this replication scheme of the DNA. I mean how the DNA will be replicated. Now let's focus on the mechanism of action of this fluoroquinolone. What actually fluoroquinolone do? Fluoroquinolone will passes they will passes from the porine channel, or they passes through the porine channel, and from the porine channel they passes the periplasmic space, and then they reaches to the target area, means they reaches to this enzyme. And to this enzyme I didn't draw the structure of the fluoroquinolone. Fluoroquinolone will be look like in this form. Fine. So this is a simple structure, fine, not a complex structure. This is simple structure of the fluoroquinolone. At position number 6 there is a fluoride group, so that's why we call that fluoroquinolone. Fine. So this is the structure of fluoroquinolone. This fluoroquinolone will be bound to that joining site. So if once these drugs will bind to this joining site, so cleavage process, although cleavage process is present, but this joining activity will be lost, or this joining function is lost. What does it mean? It means that when this enzyme is come and they will cleave, but they will not join. Why? Because here the drugs are bind. So when the drugs are bind, so totally the function is changed. How? This enzyme will just degrade, this enzyme will just cleave, and due to this cleavage DNA degradation will be occur. So once the DNA degradation will occur, so then the cell will be, so let's suppose this is the cell. Inside the cell this is the DNA material. Fine. So this DNA material, what actually happen? This DNA material will be degraded by this enzyme, but they are not resealing or joining by this enzyme. So after that this DNA will be die. After that this this cell will be die. Form this cell will be die. So that's why we also call that this fluoroquinolone is actually bactericidal in nature. Bactericidal. Fine. So this fluoroquinolone is actually bactericidal nature. Fine. Because they can kill the cell. This is a bacterial cell, let's suppose. So they will kill the bacterial cell. Fine. So that's all about the fluoroquinolone mechanism of action. Fine. Now, what are the antibacterial spectrum of these drugs? Means what are the broad spectrum activity? These drugs show best result, or these drugs are activated against Staphylococcus species, Streptococcus species, Pseudomonas species, Enterobacteria, and Enterococcus species. Against these species, these are the very important drugs, right? So these are very important drugs against these species. So that's all about the fluoroquinolone, their mechanism of action, some important discussion about the DNA replication, and the special drugs which are used in fluoroquinolones. Fine. I hope this video helpful. If still you have any kind of question in your mind, so write it into a comment. I will try my best to give you answer as soon as possible. So if you like this video, please hit the like button, share this video, and also subscribe the channel to get more interesting video lecture like that. Thank you so much.